Welcome to Study Loop.
Which one of these things is different or similar between eukaryotes and prokaryotes when it comes to gene expression? So real quick, uh, let me work with blue because that is the color I've used the least. In prokaryotes, you have a circular genome, right? And I've only drawn one gene here, but there would be multiple genes, right, across the circular genome. So every few thousand nucleotides, you would have a genome. What I've shown here in green is the gene, and in red is the promoter or operator that regulates that gene. And the RNA polymerase and all the transcription factors usually bind to the promoter, and in prokaryotes, while the RNA is being transcribed, a ribosome can attach to the RNA and starts translating the RNA into protein before the RNA is even being finished transcription. So transcription and translation can occur at the same time. So while this RNA is still finishing making the RNA, the ribosome can start translating that RNA. Okay, and there's a promoter that can regulate the rate of transcription. In eukaryotes, you have linear genes, or we have linear chromosomes, not circular chromosomes. And we also have promoters in eukaryotes. And that gene goes through transcription. I haven't drawn the RNA polymerase here, but RNA polymerase and all transcription factors would bind here and they would transcribe the gene, resulting in a premature mRNA. The premature mRNA then gets added 5' cap, a poly A tail, a polyadenine tail. It undergoes splicing where you remove introns or exons you do not need, and RNA binding proteins bind to the RNA, and all of these are different methods of affecting RNA stability, and all of these happen in eukaryotes. And then after all of that processing of the pre-mRNA, you end up in a mature messenger mRNA, and then that can be translated into a protein, and then the protein can be further modified, or its action can be further modulated by addition of chemical groups like phosphates, and those are post-translational modifications. So that's a high view overview of prokaryote and eukaryote gene expression regulation. So which one of these is found in both 3' poly A tails? They are found on eukaryotes, but not in prokaryotes. So in prokaryotes, as soon as the RNA starts being transcribed, it also starts being translated, right? So there's no step in between. There's no time for the RNA to even be modified through splicing or addition of anything. So poly A tails are not added in prokaryotes. So that is not a common thing. The same reasoning goes for 5' caps. Okay, so the correct answer is C, which is promoters, because we see that promoters are present in both prokaryotes and eukaryotes, and that is the region of the DNA to which transcription factors and RNA polymerases bind. And choice D is introns, and that is also not common in both because there is no splicing in prokaryotes. In eukaryotes, you have a division of nucleus and cytoplasm, and in the nucleus, you can process the mRNA into mRNA before the mRNA goes into the cytoplasm to be translated. In prokaryotes, you do not have the division. The ribosome is right there. As soon as the RNA starts being produced, it can start being translated. So there's no time for splicing out of introns. So the correct answer is C. Promoters is what's common between prokaryotes and eukaryotes.